Hello everyone, how are you all? Good. I hope you all are doing great. So this is your educator Chinmay. I'm from Odisha. I have completed my graduation and post-graduation from Ravensa University in subject botany as my major. So in today's session we are going to study about DNA replication. In case you want to know more detail about me, this is a brief about me. You can have a look. This is a brief about me. So in today's session, we are going to study about DNA replication. Now, my first question is, what is DNA replication? So when I'm saying replication means making a copy of certain thing, making a replica. Making a replica of DNA or making a copy of DNA is what called DNA replication. And again, we have already read in our lower grade that DNA replication is semi-conservative in nature. When I'm saying semi conservative in nature, मतलब semi conservative मतलब नाम से पता चल रहा है. When I'm saying conservative मतलब हम बोलते हैं ना पुराने ख्यालात वाले. Okay, so conservative means like पुराने ख्यालात, जिनका ख्यालात थोड़ा सा मतलब पुराना है उन्हें कंजर्वेटिव बोलते हैं. अगर हम बोलते हैं सेमी कंजर्वेटिव मतलब कंप्लीटली पुराने ख्यालात वाले नहीं हैं, थोड़े थोड़े पुराने ख्यालात और थोड़े थोड़े नया सोच उनकी, मतलब इट्स अ मिक्सचर ऑफ ओल्ड थिंकिंग, ओल्ड थॉट एंड न्यू थॉट्स. ओके, सो सेमी कंजर्वेटिव, so you can guess from the name, one of the parental strand is retained and a new daughter strand will be formed. Okay, so there is a parental strand and there is a daughter strand. सो सेमी कंजर्वेटिव मतलब पूरे के पूरे पुराना ख्यालात नहीं रखते हैं, थोड़ा थोड़ा पुराना ख्यालात, उसके साथ साथ थोड़े थोड़े नया ख्यालात, मतलब न्यू एंड ओल्ड थॉट. इट्स अ मिक्सचर ऑफ न्यू एंड ओल्ड thoughts. Hence, it is called semi-conservative. So, there is a parental strand and there is a daughter strand along with it. Okay, so if you can recall the structure of DNA, what is the structure of DNA? DNA is deoxyribonucleic acid. Why it is called deoxyribonucleic acid? Deoxy means devoid of oxygen. There is no oxygen at the 2' position. Now 2' position means what I'm talking about. So what are the constituents of DNA? You can see here only I have written DNA consists of what? There is a pentose sugar, there is a phosphate group, there is a nitrogenous base. Nitrogenous base are further classified into purine and pyrimidine. Purine further classified into adenine and guanine. What are coming under purine? If someone will ask you, what are coming under purine? Adenine and guanine. These are the nitrogenous bases. And what are coming under pyrimidine? Cytosine, thiamine and uracil. Uracil is coming in RNA. Uracil is not present in DNA. So if you look at the structure of a DNA, it consists of a pentose sugar, there is a phosphate group and there is a nitrogenous base. Nitrogenous base can be adenine, guanine, cytosine, thiamine, anything. Uracil is not there. Uracil is present in RNA. So I hope it is clear. Now, who was the one to discover the structure of DNA? Okay, who was the first person to discover the structure of DNA? Everything is written here. So if you look at the structure of DNA, it is a double helical structure, and that structure was given by Watson and Crick in the year 1953 based on X-ray diffraction data produced by Wilkins and Franklin. Okay, so how Watson and Crick gave this structure of DNA? Okay, by studying some data which was produced by Wilkins and Franklin. Okay, I hope it is clear. Now just have a look. So we already know that DNA consists of a pentose sugar, there is a phosphate group and there is a nitrogenous base. Now the question is how the nitrogenous base is connected to the pentose sugar. So nitrogenous base is connected to the pentose sugar with the help of a bond. That bond is called glycosidic bond. So this slide is very important and you can expect question from this slide in each and every examination. So you can see there is a pentose sugar connected to the nitrogenous base. Nitrogenous base can be purine or pyrimidine. It can be, if I'm saying purine, it can be adenine or guanine. If I'm talking about pyrimidine, it can be cytosine or thiamine. It can be any four of them, adenine, guanine, cytosine or thiamine. Okay, so sugar is connected to the nitrogenous base by a bond. That bond is called glycosidic bond. And sugar and nitrogenous base together, they are called nucleoside. Okay, and when a phosphate group is attached to that nucleoside, it is called nucleotide. Now the question is how the phosphate group is attached to the nucleoside. The bond so formed is called phosphoester bond. And when one nucleotide is connected to another nucleotide, it is called dinucleotide. Okay, so one nucleotide is connected to another nucleotide with the help of a bond that is called phosphodiester bond, and more than two nucleotides connected to form a polynucleotide. Hence, if you look at the structure of a single-stranded DNA, it is a polynucleotide where many nucleotides are connected with each other by phosphodiester bond. So this bond is very important. You have to remember that. You have to keep in your mind. Okay, how they are connected. How sugar is connected to nitrogenous base, how nucleoside is connected to phosphate, how one nucleotide is connected to another nucleotide. Okay, and so two polynucleotides connected with each other by base pairing. Base pairing means we are talking about hydrogen bond. How the base are paired with each other, there is a hydrogen bond. A is paired with T, there are two hydrogen bonds, and G paired with C, there are three hydrogen bonds. So they form a double-stranded DNA. Okay, so we know that DNA is a double helical structure. So these are the bonds which are playing a vital role in the formation of a double helical structure of DNA. Okay, now just have a look. This is a simplified view. You can see that it's a double helical structure and two strands, they are anti-parallel to each other. When I'm saying anti-parallel, what do you mean by anti-parallel? Just have a look. So this is parallel and this is anti-parallel. Parallel means two lines, if they are running in the same direction, then they are called parallel. Okay, and anti-parallel means two lines, they are running in opposite direction, then they are called anti-parallel. So if the polarity of one strand is 5' to 3', then the polarity of other strand will be 3' to 5'. Hence, DNA is a double helical structure and the two strands, they are anti-parallel to each other. And is this anti-parallel nature needed? What is the need? Anti-parallel क्यों है? So that they can form this base pairing. Okay, so now just have a look. This is the structure of a DNA. Okay, you can see this one is 5', this one is 3', and the other strand is here. This is 5', this is 3'. So the polarity is opposite. Okay, and you can see A pairs with T, there are two hydrogen bonds, and G pairs with C with three hydrogen bonds. So this is one nucleotide, this whole is another nucleotide, this whole is another nucleotide, and this whole is another nucleotide. So one nucleotide is connected to another nucleotide with phosphodiester bond, and this pentose sugar is connected to this nitrogenous base by glycosidic bond, and this whole nucleoside is connected to this phosphate. Nucleoside is connected to this phosphate with the help of phosphoester bond. Okay, I hope it is clear. Now have a look. This is the structure of DNA. You can see, I hope now you can easily understand what is actually happening. So it's a double helical structure, double helical. Why I'm saying helical? It is like a helix. It is like a helix. Hence it is called double helical structure. Two strands, they are anti-parallel to each other. The polarity of one strand is 5' to 3', polarity of other strand is 3 to 5, and bases are attached to each other by hydrogen bond. A pairs with T by 2 hydrogen bond and G pairs with C by 3 hydrogen bond. Okay. Now, in this session, we are going to study about DNA replication. So before understanding DNA replication, you need to understand certain terms. Okay, in DNA replication, we have heard this term a lot more time. Supercoiling. What do you mean by supercoiling? Okay, coiling. I think all of you know what is coiling. And when I'm saying supercoiling again are of two types. One is positive supercoiling, one is negative supercoiling. So when I'm saying positive supercoiling, just have a look. Suppose you can see this is a DNA, and you can see how many turns are there. 1, 2, 3, 4. So there are total 4 turns. Okay, so now you can see this is another DNA, and you can count the number of turns. 1, 2, 3, 4, 5, 6. So here the number of turns increased. Okay, so this is the initial number of turn, and this is the final number of turn. Okay, if I will find out the difference between these two, so final minus initial, what I'm going to get? I'm going to get +2. So positive, the value is in positive, hence it is called positive supercoiling. But here you can see initially the number of turn is 4, and finally you can see the number of turn is 3. Okay, so what is happening? If you are going to find out what is the difference, then you can see the difference, like final minus initial, the difference is -1. So the value is in negative, hence it is negative supercoiling. I hope it is clear. What is positive supercoiling? What is negative supercoiling? Positive supercoiling कब होगा? जब number of turns बढ़ेगा, उसे हम बोलेंगे positive supercoiling. जब number of turns घटेगा, उसे हम बोलेंगे negative supercoiling. एंड ऑलवेज रिमेंबर, जब हम डिफरेंस कैलकुलेट करते हैं हमेशा फाइनल से इनिशियल करते हैं, इनिशियल से फाइनल नहीं. फाइनल से इनिशियल आपको सब्सट्रैक्ट करना है, then only you are going to get the difference. If the difference value is in positive, then it is called positive supercoiling. If the difference value is in negative, then it is called negative supercoiling. I hope it is clear. So what is supercoiling, and what is positive supercoiling, and what is negative supercoiling? And which enzyme is responsible for removing the supercoiling, we are going to study about it. Now just have a look. So this is the whole DNA replication process. Okay, this is the whole DNA replication process. As you can see, just remember one thing. This is a very simplified diagram that I have drawn. So just try to understand the whole concept, like what is happening. So DNA replication, we cannot start DNA replication randomly at any site. There is a particular site. From that only the DNA replication will begin. So that particular site is called ORI, origin of replication. And there are some enzymes, there are some proteins, they form a complex. And what is their function? Their function is to find out the ORI site. Their function is to find out कि कहाँ से replication start करना है. They have to, their function is to find out the ORI site, origin of replication site. So what they are going to do, they will search for that site. वो लोग ढूंढने लगेंगे, एंटायर डीएनए में वो सर्च करने लगेंगे कि वो जो ओराई साइट है वो कहाँ पे है. एक बार उन्हें वो ओराई साइट मिल गया तो वहां पे आके वो बैठ जाएंगे. ओके, फिर नेक्स्ट स्टेप क्या है? नेक्स्ट स्टेप में क्या हुआ? There is another enzyme called helicase. Helicase का काम क्या है? The function of helicase is to break the hydrogen bond. You all know that the bases, the nitrogenous bases, they are connected with each other by hydrogen bond. So the function of helicase is to break that hydrogen bond. Okay, because for DNA replication we need a single strand, because the single strand will act as a template. Double strand can't act as a template. We need a single strand for replication. ओके, सिंगल स्ट्रैंड कब फॉर्मेशन होगा? जब हाइड्रोजन बॉन्ड, दो स्ट्रैंड के बीच का जो हाइड्रोजन बॉन्ड है, जब वो टूटेगा तब हमें सिंगल स्ट्रैंड मिलेगा. तो हेलिकेज का काम क्या है? यू कैन लाइक इमेजिन, आई हैव ऑलरेडी ड्रन इट. इट इज जस्ट लाइक ए सीजर. उसका काम क्या है ना? हाइड्रोजन बॉन्ड को ब्रेक करना है. ओके, सो नाउ what is the first thing? ये है entire DNA. You can see this is the actual structure. Okay, this one I have drawn for your easy understanding. So this is the whole structure of DNA, and we cannot start the DNA replication randomly at any site. There is a particular site from which we can start the replication. So that particular site is called origin of replication, ORI site, origin of replication site. So what is going to happen? The origin recognition complex, there is a complex, they are going to search for that particular site where the replication will start, ORI site. Okay, once they found that particular site, the rest of the enzymes will come to that place and they will carry out the next steps. Okay, so helicase is the one which is responsible for breaking the hydrogen bond. And we need a helicase loader. Okay, helicase cannot get loaded to that particular site of its own. So we need a helicase loader to load that particular helicase enzyme onto that particular site. What site I'm talking about? ORI, origin of replication. So now you can imagine in this way, if there are wires in your house. Now, okay, just imagine like two intertwined wires are there. If I'm trying to open it from the middle, so you can imagine at the either end, the number of supercoiling, means the number of coiling will increase. Okay, so number of coiling will increase. You can see here the number of turns are 1, 2, 3, 4, 5, 6, and 7. But you can see here the number of turns has increased. This is 1, 2, 3, 4, 5, 6, 7, 8. Okay, so when I'm trying to open it from the middle, the number of supercoiling or the number of turning will increase on either side. So as a result, what will happen? We need another enzyme to remove that supercoiling. So that enzyme is gyrase or topoisomerase. Again, topoisomerase are of two types. One is topoisomerase 1 and topoisomerase 2. You can see what is the function of topoisomerase 1. Topoisomerase 1 is going to break only one strand. Okay, cleave only one strand. You can see what is happening, how the enzyme is functioning. You can see the topo 1 is cleaving one strand of the DNA, and you just have to turn it. Okay, cleave it and turn it. So as a result, what will happen? They are removing the supercoiling. They are simply removing the supercoiling. Okay, it is very easy. You can imagine in this way, like two wire intertwined with each other. I'm cutting one strand. From that two, I'm just cutting one strand of the wire or one wire and turning it. Okay, so you can see the number of turns here. The number of turns is decreasing. Okay, so that is the function of topoisomerase or gyrase. Gyrase in case of prokaryote and topoisomerase in case of eukaryote. Okay, so these are the enzymes responsible for removing the supercoiling. Okay, and there are some single strand binding protein. What is the function of single strand binding protein? You can see topoisomerase, they are responsible to remove the supercoiling, and helicase is responsible to break down the hydrogen bond. And after breaking the hydrogen bond, we are going to get a single strand of DNA. Okay, we are going to get a single strand of DNA. Okay, so this is the whole thing. You can see a bubble-like structure, and this is what called a fork. Okay, this is what called a fork, bidirectional fork. Bidirectional means if the replication is proceeding in both the direction, then it is called bidirectional. If it is proceeding in one direction, then it is called unidirectional. Okay, so this is the structure of a fork, and this structure is what? This is helicase. Helicase is hexameric. Hexameric means made up of 6 subunits. Okay, you can see this is a helicase. This is the structure of a helicase. Okay, so now single strand binding proteins are there. What is the function of single strand binding protein? एक बार डीएनए का जो 2 स्ट्रैंड है एक बार ऐसे सेपरेट हो गया, बहुत सारा चांसेस है, बहुत ज्यादा चांस है कि वो फिर से ऐसे रीएनील हो जाएंगे. तो सिंगल स्ट्रैंड बाइंडिंग प्रोटीन का काम ये है कि वो आके ये जो सिंगल स्ट्रैंड फॉर्म हुआ है उसके साथ बाइंड हो जाएंगे और उसे बोलेंगे कि नहीं नहीं फिर से तुम नहीं जुड़ सकते, क्योंकि तुम्हें ऐसे ही रहना है सो दैट आई कैन कैरी आउट द रेप्लिकेशन इजीली. तुम फिर से जुड़ गए तो मुझे फिर से मतलब एनर्जी लगेगा हाइड्रोजन बॉन्ड को तोड़ने में. तो सिंगल स्ट्रैंड बाइंडिंग प्रोटीन आके जुड़ जाएंगे और इसे क्या करेंगे, वो जो स्ट्रैंड सिंगल स्ट्रैंड बना है उसे उसी पोजीशन में रहने देंगे. दे प्रिवेंट द रीएनीलिंग ऑफ द स्ट्रैंड. ओके, दे प्रिवेंट द रीएनीलिंग ऑफ द स्ट्रैंड. ओके, so now what is happening next? You can see now there's another enzyme called DNA polymerase. DNA polymerase 3. Okay, so DNA polymerase 3 is playing a vital role in elongation. Okay, so there are 3 DNA pol in case of prokaryote. DNA polymerase 1, 2 and 3. All 3 are playing a vital role, but DNA pol 3 is playing a vital role in elongation. So what is happening? DNA polymerase 3 cannot initiate the replication part. Okay, it can only elongate, it cannot initiate. For initiation we need a primase enzyme. What is the function of primase enzyme? The function of primase enzyme is to add a primer. Okay, and this primer is a RNA primer. And one thing you need to remember, that this primase enzyme, what they are going to do, primers should always be added at the extreme end. Okay, so just have a look. This one is 5 prime to 3 prime, so I can easily add a primer here. There is no issue. Why? Because DNA pol 3 enzyme can only elongate the 3 prime end of the strand. It cannot elongate the 5 prime end. It can only elongate the 3 prime end. So in this case, I'm not going to face any kind of issue in this strand, in case of this strand. But in case of this strand, if I'm going to add a primer here, you know that 2 strands, this would be anti-parallel to each other. So when I'm adding a primer here, what will happen? When I'm adding a primer here, what will happen? It will be like 3 to 5. And the problem is that DNA pol 1, in order to elongate, it needs a template, right? It needs a template. And there is one more thing. It can only elongate the 3 prime end. It cannot elongate the 5 prime end. It can only elongate the 3 prime end. And if DNA pol 3 will try to elongate this strand, how it will elongate? Because there is no template in this side. There is template in this side. And DNA pol 3 cannot elongate the 5 prime end. It can only elongate the 3 prime end. Okay. So as a result, we are going to face a little bit difficulty while elongating this strand. Okay, while replicating this strand. This strand, there is no issue. But in this strand, we are going to face a little bit difficulty. So we have to wait. We have to wait for a certain time. Okay, then only the replication or the elongation of this strand will begin. And you can see this is the thing. Here, here we are adding only one primer, and that is enough. Okay, but in this case, we have to add several primer. These are the primer you can see. We have to add several primer. Okay, so the fragments so formed are called Okazaki fragment. Okay, it is a lagging strand. This strand is a leading strand and this one is a lagging strand. Why? Because you can see the fragments so formed are very like small. These are fragment. Okay, these are fragment. These are small parts are there. Okay, so it's a lagging strand. Okay, because this strand is forming later. This strand is forming earlier, hence it is called leading strand. And this strand is what? This strand is lagging strand. Why? And because it consists of Okazaki fragment. And here we need several primers to carry out the function. Okay. So in a higher class, we are going to study a little bit more detailedly about it. For now, just remember one strand is leading strand and one strand is lagging strand. Okay. Now there is one more thing, this primer, they are made up of RNA. So this primer, they need to be removed. Once the elongation is done, this primer, they need to be removed. And the removal of the primer is done by another enzyme. The name of that enzyme is DNA pol 1. DNA pol 1 is responsible for primer removal and gap filling. Okay. So now just have a look. Termination. So in termination, the termination stage, it is different in case of prokaryote and in case of eukaryote. Because in case of prokaryote, the DNA is usually circular one. But in case of eukaryote, it's a linear one. So in case of termination, the steps are a little bit different. Okay. So you can see in case of prokaryote, it's a circular DNA. So you can have a look. In a higher grade, you are going to study more. For now, I'm just giving you a brief idea what is happening. You can see, suppose just imagine the replication is taking place in both the direction, in this direction also in this direction, because it is a bidirectional one, so it is taking place in both the direction. So just imagine like if you are approaching in this direction, then you are allowed to go. But if you are approaching in this direction, you are not allowed to go. Because this two fork, they need to meet at a particular point. There's two forks. Suppose this fork is moving, one fork is moving in this direction. Okay, and one fork is moving in this direction. These two forks, they need to meet at a particular point. Otherwise, it will create problem. Okay, so if the fork is moving in this direction, it can proceed. But if the fork is moving in opposite direction, it cannot proceed. So this fork will stop at this point. This fork will move in this direction and they will meet at a particular point. Okay, they will meet at a particular point. And once that is done, DNA replication is done. You can see it's a ring-like structure. It's like this. It's like this. And we need to separate it. Okay, how the separation will be carried out? Okay, the decatenation. This is called catenated DNA. So decatenation is carried out by another enzyme. The name of that enzyme is topo. Okay, so this is how the termination occurs in case of prokaryote. But in case of eukaryotes, the things are a little bit different. Okay, in case of eukaryote, there is a telomerase enzyme. That enzyme is responsible for providing a template, and we need to elongate that particular strand. Then we can remove the primer. So there is a lot more whole step involved in it. Okay. This is how DNA replication takes place. There is initiation step, there is elongation step, and there is termination steps. And the steps are common in both prokaryote and eukaryote. The only difference is in prokaryote, it's a circular DNA, and in case of eukaryote, there is a linear DNA. Okay, so I hope everything is clear. Yes, I hope everything is clear. Just have a look. Just a second. Yeah, so now just have a look. These are the enzymes and their function that we have already discussed. DNA polymerase 3 is responsible for elongation, pol 2 is responsible for repair, 1 is responsible for gap filling and primer removal, helicase is responsible for breaking the hydrogen bond, topoisomerase and guide is responsible for removing supercoiling, and single strand binding protein, they prevent reannealing. Okay, and primase is responsible for adding the primer. So I hope you all enjoyed my session. I hope everything is like you understood the whole concept. Okay, if you have any doubt, you can ask me in the doubt box. Till then, please take care. Bye everyone.